overconfident, maybe. So we have formed a panel of experts, and today vaccine and vaccination is presented by this panel, moderated by none other than Dr. Vineet Sehgal, sir. And I asked Dr. Richa Prashar, our Honorary Secretary, to introduce the panelists and Dr. Vineet Sehgal.
So we have formed a panel.
Yeah.
To start today's session, panel discussion, I welcome Dr. Vineet Sehgal, who's a senior pediatrician and pulmonologist in Max Hospital, Shalimar Bagh, to moderate the session. And I would like to just clear that I request all the participants to please mute themselves while the discussion is going on and to put up their questions in the chat box, which will be taken up. And I request Dr. Vineet Sehgal to please introduce the other panelists. Over to you, sir.
Thank you, Dr. Richa and Dr. Gaba, for giving us the opportunity to hold this panel discussion on COVID-19 vaccines and vaccination. As we were just discussing a few minutes back, there is a huge load of information available out there, which does not necessarily translate into knowledge. Everybody has an opinion. So we'll try and kind of sift through all the information and present the important bits and pieces and conclusions to you, which are useful on a day-to-day basis and relevant to us as pediatricians. As far as today's panelists go, let me introduce Dr. Chugh, sir. First of all, he's a towering personality both in pediatric pulmonology and intensive care, but beyond that, he's a very well known practicing pediatrician also, and his knowledge does not stop at just pulmonology or intensive care. And now at present, he is at Fortis Hospital, Gurgaon. He is the chairman of pediatric services there. Welcome, Dr. Chugh, sir. Thank you for sparing time. And of course, Vipul, Gaurav, and Puneet, I would introduce in one go. All the three of them are senior pediatricians practicing in West Delhi, and all of three of them are well-read and have a special interest in vaccines and vaccinology. And even in past seminars, I'm sure you have kind of come across them with their opinions and their knowledge on vaccines. So I'm sure they'll enrich the panel discussion today with their inputs. So I'll start right away in earnest, because I'm sure there are a lot of questions, plenty of answers, plenty of information. And we invite the audience to post their questions or comments into the chat box, and Dr. Richa or Dr. Neeta will forward the questions to me, and I'll take them at intervals, at 15-20 minute intervals, and try to answer all the questions. And we will also invite comments, but pertinent and to the point comment, nothing very long-winded. So to start, Dr. Puneet, I'll open the discussion with you. So first we'll talk about the vaccines, and then we'll go on to the vaccination and the strategies, etc. So a quick overview of the vaccine platforms that are available and the vaccines which are available at present against the SARS-CoV-2 virus.
Good afternoon, everyone. Thank you for giving this opportunity. Starting straight away, vaccine platforms are different approaches basically. The purpose of any vaccine is to deliver the protein, the immunogenic protein, which induces protective, that is neutralizing antibodies. And vaccine platforms are different approaches to deliver the protein or to generate the protein inside the body, in the host. So the conventional platforms are live attenuated and inactivated vaccines. Inactivated vaccine is a vaccine where the virus in question is inactivated and is delivered as a vaccine with its adjuvants. For example, in COVID vaccines, it is Covaxin, Sinovac, Coronavac. These are inactivated vaccines. Then there is the viral vector vaccine, that is Covishield. Here, another harmless virus carries the genetic code which is delivered to the antigen presenting cells, which produce ultimately the immunogenic protein, that is the spike protein in COVID-2. The spike protein is produced by the host cells, and the genetic code is delivered by the harmless virus, which is adenovirus in Covishield. So another is the RNA vaccine, where the naked RNA, as such, RNA encoding the code for the spike protein is encapsulated in a lipid core and is delivered. DNA vaccine is a DNA plasmid, which again carries the genetic code for the formation of the spike protein. It is introduced by electroporation or needle-free technique, and ultimately the protein is generated inside the host cell. Host subunit vaccine is where the protein itself is directly introduced in the body. Just like we have hepatitis B vaccine, in similar we have COVID vaccines also on this platform. The Novavax is one example. So these are all the different platforms. And with COVID, we have a special situation where we have so many platforms and so many vaccine candidates. As on January 6th, where there are more than 334 vaccine candidates available on all these platforms: RNA vaccine, DNA vaccine, vector vaccine, live attenuated protein vaccine. All these are under development. We already have 26 are in use, and around 118 are already in clinical trials.
Thank you.
So these are a quick overview. We'll discuss further in next question.
Yeah, we'll discuss them in greater detail as we go along. So, but plenty of platforms and plenty of vaccines. So it's going to be a whole plethora of choices. So, sir, Dr. Chugh, going on to you. From what we understand, vaccines used to take decades to develop, but this COVID vaccine, within a year of the pandemic, we had vaccines. So what happened which kind of accelerated this development? How come so quickly?
Well, yes, that's true that this is what was the norm. It will take many years and sometimes even decades. In fact, there is a chart which shows that it took more than 100 years for some vaccines to be made. Anyway, before we go on to say that there is anything special that we'll be telling you, let us be very clear that humility is essential in this COVID vaccine or COVID disease, because there are too many unknowns still. It's a new disease. Not many of us know many things about it. In fact, nobody knows everything about it. The precise duration of immunity, whether SARS-CoV will be a seasonal infection later, whether antiviral therapies will work, are they working, whether even more transmissible and immune evading new variants will arrive after Omicron, and are there any predictors for necessary? Well, predictors are necessary. We are trying to tell what will happen in next few weeks and next few months, but we know that there is no mathematical certainty. So these are the unknowns, and in the light of this, let's discuss what has been asked to me. Like this chart shows, there are many vaccines which took years and years to develop. And at the bottom, it's shown what has happened in few months with COVID vaccine. With COVID vaccine was ready within a few months. So that's what it is. Why? And let's discuss why and how it happened. You see, the basic thing was genome sequencing is now available. Human genome is available on the internet free of charge. So genome sequencing became available in January 20 itself, mainly because of computer technology. And China did hide a lot of things, but at least once pressurized a little bit, did release the details, and so genome sequencing became available early. Then pandemic of a severe disease with high mortality it is, and very soon it was known that it's something which has a high mortality, and it's spreading very fast, very, very fast. So that induced a lot of public fear. And once there is public fear, political pressure arises, and our politicians are smart. They know how to make the best of the situation. So they decided to fund private companies, especially in US, where lots and lots of funds were given to these two manufacturers, Moderna and Pfizer, and many more. Similar trend followed in the rest of the world, including India. And then there was previous research on SARS and MERS vaccines had been done in the last 20 years or so, where we had a base. We knew a lot about it, but those vaccines did not see the light of the day for various reasons. Now, unlocking the coronavirus secrets, well, that also happened quickly, as much as was required for making a vaccine at least. Of course, we continue to learn more about it. Then there is a finding of a delivery system. Now, when, as I explained in the previous slides by Dr. Puneet, what we have to see is that an antigen has to be delivered to the tissues in human body. For that, mRNA and adenovirus are the two main delivery systems that were for which there was a base already. There was a lot of knowledge about it. So benefiting from the past research on these two, the scientists started making the vaccine. Then when it comes to recruiting volunteers, that also happened very fast. Results came fast, and jumping the line at regulatory agencies, FDA and so on, also ICMR included, did a good job. Now, mRNA platform had just been developed, it had been developed quite a few years back, but it really matured very recently. Researchers knew from their work on MERS and SARS that it was the best to tune the RNA sequence to stabilize the resulting spike protein in the form it adopts before it docks with the host cell. So just before it enters the host cell, RNA has this particular sequence and particular shape and form. If we freeze it there and trap it there and then deliver it through the mRNA platform, this is what will work best. Now, scientists had already built these rapid genetically based templates largely because of ongoing research against HIV. So HIV has been here for quite a few decades, and the research on that also was incorporated or taken up from there into our COVID-2. Now, one of the templates directly carries a non-functional partial strand of a viral mRNA to cells in the body, delivering instructions for those cells to create copies of that protein that immune system recognizes as foreign. That's what was used by this Pfizer and Moderna shots. This is called, in a slang kind of way, plug and play vaccine manufacturing. That is, it's a real quick method. Decades of trying to attack HIV has created a library of safe weapons to use quickly against any new would-be viral infections. Now, scientists are very optimistic that come any new virus and they would quickly be ready. Now, the mRNA for the coronavirus spike proteins gets packaged inside tiny bubbles, as was told to us in the previous question answer, that lipid nanoparticles are used. These tiny fat bubbles are solely used for dozens of other drugs, some approved and others still in the pipeline. Now, so all that needed to be changed was to target SARS-CoV-2 inside the lipid package. Those fat bubbles are so crucial because if the mRNA is injected alone outside a bubble directly, it breaks down quickly, degrading into harmless biological raw bits and pieces that get recycled by our bodies without initiating an immune response. The contents of this fat bubble are called the payload, just as you would say for a satellite. Now, AstraZeneca, so we know that it's a vector-based vaccine, and research on selection of vector adenovirus had been going on for years. It's cheaper than mRNA vaccines. Now, slowest part of the vaccine development in the past has been testing in labs, in animals, and then in 3 phases in humans. That's the cycle that any vaccine normally takes. Each stage means escalating costs. With billions available to companies, they could take the risks. All these stages were gone through with COVID vaccine also. But alongside, manufacturing had started. You know that Serum Institute had manufactured the vaccine well before the trial was, in fact, just started, and they had manufactured the vaccine in large quantities. So large number of cases were going on when the epidemic and the pandemic had started. So that meant that quickly we could recruit lots of patients. For instance, it took just under 16 weeks to recruit and enroll more than 43,000 volunteers for final phases of testing Pfizer vaccine. And some of us who were a part of the previous vaccines, for example, Prevenar, we used to enroll 2-3 patients in a month and per site. It was a slow process. Here, there are patients and patients around. It was so simple. Volunteer recruitment began for clinical trials of rabies mRNA vaccine. For example, there's a comparison with rabies. in 2013. It took 813 days to get 101 participants enrolled. Based on this comparison, that's roughly 730 days were gained for COVID, 2 years. Now, scientists can begin to calculate a vaccine's efficacy when a sufficient number of people in the group that got a placebo rather than a vaccine have gotten infected naturally. If an outbreak petters out, as had started happening in the middle when the first wave was over and the second was not yet around, it takes, in that phase, those vaccines who started enrolling took much longer to enroll the numbers that they had decided. That's also true if a virus naturally is spread slowly. For example, HIV, herpes, HPV, there it took much longer to enroll. And there is a comparison with HPV vaccine on this slide. HPV trials took 529 days. That is 1.4 years to reach a point at which 3.8% people in the placebo group had been infected. Pfizer vaccine phase 2 and 3, it took much smaller number of days, 105 only. That means 424 days were gained compared to HPV. Why? Because just months into pandemic, SARS-CoV-2 was infecting hundreds and thousands of people around the world. Now, SARS-CoV-19 virus does not mutate as fast as influenza virus. That's another additional. It does not foil the human immune system, unlike HIV. It does not block antibodies like herpes. So we had a virus also, which in some sense, of course, devastating, but was amenable to quick vaccine development.
So
conventionally, we had live attenuated, inactivated, and subunit vaccine.
I'll interrupt you here.
Sorry.
So basically, we had the technology in place, luckily, and we had governments which were willing, and we had industry who stood to make billions, so they were ready to put in the millions. And of course, we were lucky. It's actually unlucky, but we had large number of subjects readily available. So everything kind of came into place, plus the regulatory authorities hastened the process and all the files and probably the red tapeism, etc. So we had vaccines, and more vaccines are coming in. So within, I would say, 2-3 years, we would have multiple vaccines. So Dr. Vipul, we've already, I think Puneet and Dr. Chugh have already talked about the different vaccines, but just a little more of detail that, these newer vaccines, which we really don't understand, viral vector vaccine, mRNA, DNA. For us, it used to be a live vaccine or a attenuated vaccine or a killed vaccine. So what are these new platforms that we are talking about?
Thank you, Dr. Vineet. As you have already written that we already had live attenuated and inactivated vaccines and some of the subunit vaccines before COVID vaccine came into play. After this, now we have so many vaccines with these newer technologies. For example, we have 3 major technologies. One is viral vector technology, under which we have AstraZeneca vaccine marketed as Covishield. We have Russian vaccine by the name of Sputnik V. We have Johnson & Johnson vaccine with us. How it works is, next slide, please. How it basically works is, you have a virus, you take out the viral protein or the antigen, which is primarily responsible for the virulence of the virus. Here you call it spike protein because it is spiked on the plasma membrane of the virus. And you know this spike protein, and you know the genome of this spike protein, as Dr. Chugh pointed out, and you mount this genome, recombinant genome, onto a live virus, which is adenovirus. It acts basically as a agent to carry this genome into the human body, and basically body consumes this adenovirus. It acts as a vector, it carries, and body consumes this adenovirus, and this adenovirus is processed inside the bodies through B and T cells, producing antibodies, memory B cells, and memory T cells. This is how it works. Why adenovirus? Because adenoviruses are common viruses that human beings encounter normally, and it is antigen presenting cells that are there in our body identifies them well, and chances of missing them, not identifying them, and not taking up are very less. That is why adenovirus was chosen. Here in this case, it's a chimpanzee adenovirus which was taken up and used. This principle applies to all three viral vector vaccines. Now, second, the most talked about or most commonly said to be the most sophisticated vaccine, the mRNA vaccine. But here what you do is you create a synthetic mRNA of the spike protein, and you inject that into the human body. And when this synthetic viral mRNA is taken up by the body and enters into the human cell from the ribosomes, multiple copies of this mRNA are made, and they help the human protein to be converted to be made to similar to SARS-CoV-2 viral protein. And those SARS-2 viral proteins, they basically gets consumed by the body and produce antibodies, and they are taken up by the T and B cell system inside the body. Now here in this picture, I wish to just compare how these viral vectors and mRNA vaccines are basically different. In first, we are injecting the spike protein through a adenovirus, through a vector, and that vector is producing mRNA inside the body, and that mRNA is taken up by the ribosomes and further immune response is created. But in mRNA vaccine, this message or this mRNA is directly injected, taken up by the ribosomes, and then further immune responses being created. This also explains why mRNA vaccine is a step ahead viral vector vaccine. Next, please. No, there is another group of vaccine which is called DNA vaccine. What you do is you take the genome of spike protein and you encode that genome onto DNA and inject inside the body. Now, these DNAs are taken up by antigen presenting cells, whether they are dendrite cells or whatsoever, macrophages, dendrite cells, they are taken up by them, and they are processed inside the body through T cells and B cell system, giving rise to humoral and cellular responses. So this is comparatively easy to do in comparison to mRNA and viral vector vaccines, but it needed a lot of research, and currently we only have one vaccine commercially available and one vaccine in pipeline, which is using this technology. Next, please. Now, this technology of subunit vaccine was already there, but what new has been added to this subunit vaccine technology is the nanoparticle. Lipid nanoparticles, basically you need less amount of antigen, and spike protein can be easily, as Dr. Chugh said, plug and play. If you have new virus coming in next few months and you catch up the new genome, you can easily change the spike protein from existing spike protein to a new spike protein. And if you need a new vaccine in a very short time, it becomes very easy to create a subunit vaccine. And subunit vaccine basically is a very less amount of spike protein bound with nanoparticles. And when you inject it directly, these spike proteins get consumed by the body, and again, humoral and cellular responses take place inside the body.
Okay, so again, lot of information, and it will take time to digest all that. But from what I can make out is that we're in a good space as far as if new mutants or variants come along, we have the technology to switch the production to the more appropriate variant, and we can rapidly produce new vaccines if needed. But Gaurav, I'll bring you now into the discussion and ask you that, we all talk about the spike protein, but what I understand, would it not be better if we could form antibodies to the entire virus particle and not just concentrate on the spike protein? So wouldn't a whole virus vaccine be better since it has all the antigens, covers all bases, so to say?
So thank you, Dr. Vineet. So coming to the whole virus vaccine, I think we've all kind of grown in our practice using measles, MMR, chickenpox vaccine, and having confidence on the live attenuated viral vaccines. But in the case of so whole virus, we can have either a live attenuated vaccine or an inactivated vaccine. Live attenuated would get a good immune response across all cell lines, including the humoral as well as cellular immune response. But then the risk of vaccine-induced disease would be there, and it cannot be used in immune compromised in pregnancy. And so those are limitations. Plus, for the inactivated vaccine, I think they would be stimulating basically the humoral immune system and not get a very good cell-mediated immune response. But across both the formats, we would need a large amount of virus to be grown and either attenuated or inactivated. And that would entail a lot of cost as well as a risk of the virus kind of leaking out. And the newer technologies, they were already well developed and they were cheaper to kind of upscale faster, and a better immune response is expected with them. The inactivated vaccine would have a 10-fold lower antibody response to the other technologies, as we are seeing with the 2 vaccines that are available to us as well. So I think that ways, although the whole cell vaccine would be, the inactivated vaccine would be much safer and much more stable, but then the immune response would not be as good as is expected when with the newer technologies.
So as far as SARS-CoV-2 virus goes, I guess the whole cell vaccine is kind of out from what I can make out. So we go back to our other platform.
Not a very encouraging platform to be working on.
Sure.
Just to add one point, Dr. Vineet here. See, COVID-19 was a new virus, and epitopes in COVID-19 virus were not there with the humans at this point of time because we never had encountered this virus before. So acceptance of the whole cell vaccine as such in a human being would have been less. There would be many misses. Many people, many bodies would not identify the whole virus as such. And especially the first dose would be just the priming of the human body and would be missing to identify the virus as such. So that is the biggest drawback, and that is the most common reason what Dr. Gaurav said, that it has 1/10 of the antibody response and not being recognized.
In fact, there is another
it used to be called the novel coronavirus vaccine, the coronavirus novel. So that's what you're kind of alluding to, that it was a novel virus for the human immune system and it would not have reacted to the vaccine.
Yes.
I get your point. Thank you. Dr. Chugh, now we'll go on to we've talked about the technology and the type of vaccines, but now let us look at the primary endpoints, or what are we looking for in the vaccine to develop immunity. So from what we can judge over the past 1 year that we've used the vaccine, that they seem to be more effective against severe disease. All of us kind of agree to the fact that they prevent severe disease, prevent hospitalization, cut down on the need for oxygen. But the mild disease still goes on, the transmission still goes on. So is it a failure of the vaccine?
Well, in a sense, it is a yes and a no. No, because you have already said that so many advantages or benefits have been reaped already by vaccinating a huge population of this world. Because we know that a vaccine should be able to do, a good vaccine should be able to do all these things listed on this slide. It should prevent infection in the recipient. It should prevent the spread by the vaccinated to others, even if he's an asymptomatic carrier. It should prevent symptoms almost completely and totally. Then preventing, it should definitely prevent severe disease and hospitalization. It should prevent ICU admission, and it should prevent death. So if you look at each one of them, there are figures available around the world which tell us, yes, how much effective it has been in each one of these points. So when we have to look at efficacy and effectiveness, we should be very clear what we are talking about. It's important to distinguish between these two. Now, vaccine efficacy is a measure of how well the vaccine works in a clinical trial, whereas vaccine effectiveness determines how well the vaccine works in real life. And we know that there's a big difference between the two. How it is given, how it is transported, and what all actually happens, and so on. What kind of population we are covering in a trial, and what kind of population are we covering in a real life, and so on. Now, while these numbers are likely to be very similar, they may not be identical. We expect a good vaccine to be almost equally efficacious and effective. Now, next slide, please. Yeah, sorry. Okay. So if we had gone back to the previous slide, please. So if you look at the bottom 3, 4, 5 things, 4 at least, it is preventing symptoms, it is preventing severe disease, it is preventing hospitalizations, it is preventing ICU admissions, it is preventing death. But in the first 2 parts where it is supposed to prevent infection and a spread, probably it has at least been only partly effective, or we can say there is a failure, yes.
So given the horror images that we have of the pandemic, hospitals running full, lack of oxygen, people dying on the streets, I guess if you look at those, then the vaccine has been effective. But yes, all of us are getting the infection in a low grade sort of a form, and I guess we've got used to the idea that chut put to hoga, and we will recover, and hopefully the vaccine will prevent anything serious from happening. So as you said, it's a hit and a miss, but you win some, you lose some. And at the moment, we seem to be winning at least the battle against the more severe forms of the disease. So, Dr. Vipul, now with all the new strains coming in, you have these reports that the earlier vaccine will not work against these strains. So, and the virus we expect to keep mutating or keep changing. So, giving the vaccine to millions and millions and the whole effort, logistics, is it really worth it? Because 6 months down the line, you'll have another strain, then you go through the whole process again. And does the vaccine break the chain of community transmission?
I think to answer the first question, we need to understand what degree of change this virus is going through. And does the existing vaccine, which is made for Wuhan strain, is effective against the current new strains which are coming up, saying that it has 40 mutations, 50 mutations, and maybe somebody would claim to have noticed a new virus with more than 100 mutations. So we need to understand, for example, this photograph, if we look at, this is the situation when Delta came last year following primary vaccination. So there were breakthroughs with Delta strain, but it was seen that memory B cells were saving you a lot. And these breakthroughs were not severe in vaccinated population. They were being saved by ICU admissions, oxygenation, hospitalizations. They were significantly low, and memory B cells were significantly there inside the vaccinated population. So it means that vaccine works, and even if the new strain is going to come in future, there would be some savior from these memory cells. Next, yeah. Now, this is post-vaccine neutralizing antibodies against different strains following the initial, this is following mRNA vaccine. So if you look at, this is following a combination of Covovax and Covovax plus AstraZeneca vaccine. It's a Chinese study, and they found that neutralizing antibodies against different strains like alpha, beta, delta, and the wild type were significantly there in population, and they were able to neutralize these different strains. So the basic vaccine, which is made for Wuhan strain, is working for other strains also. And in this study also, this is US data, which basically suggested that even after 6 months of 2 doses, 80% Delta and 57% Omicron was vaccine was effective against these diseases. And if booster given, it is almost in tune of 90%. So with these new strains, there is definitely a protection from the primary vaccine, which was made for Wuhan strain. Now, what is the reason? Reason is 80% epitopes are same in Omicron, which has 50 major mutations. So here there is a comparison of 3 viruses around here, 3 vaccines around here listed here, and it has the T cell response, both CD4 and CD8 response is significantly good enough to save you from Omicron when you have taken a vaccine which was primarily made for Wuhan strain. So saying that new strains will come, this vaccine will not work, as of now doesn't seem to be like that. And you all remember initially we talked about plug and play vaccines, where as soon the new strains will come, we will copy the genome and plug into different kind of vaccines, both mRNA vaccine and subunit vaccines. It would be very easy to plug and play and find newer vaccines for them. Now, this is quite interesting. Somebody recently claimed that Omicron is a natural vaccine, although I would not go by this statement, but let us take this otherwise. Omicron, although has multiple mutations, but if you look at the antibodies produced by Omicron, it saves you from different other strains like alpha, beta, gamma, delta. So in a sense, it gives a message that, okay, the backbone of the virus is same. Although mutations are happening, there is a crisscross happening amongst the genes, but the whole backbone, basic structure of the genome is same as which was of Wuhan strain. There is another thing, escaping from primary vaccination, because people are taking it is in a different sense. They don't want to take vaccine because they are saying it has already mutated, further mutation, kitni vaccine loge. So no, actual disease process, if you look, it is now well known that it damages the T cells, it alters the T cells, and there is a hyperactivity of T cells following natural infection, leading on to many autoimmune disorders, long COVID and things, which is not there with vaccine. And when you get immunity from vaccine, this doesn't happen. So getting a natural infection and damaging T cells is something serious which we need to note about. Now, your last question that you asked, and this question was there in the previous question also, that when Dr. Chugh was asked, will it be effective for community transmission? I think it is too ambitious for us to ask for a vaccine which will actually stop community transmission of any virus, any respiratory virus. See, you need to have enough amount of mucosal antibodies to stop transmission, which means at any given point of time, when you encounter this virus, enough number of antibodies in your nasal mucosa to counter it immediately. So this is not practically possible. It is only practically possible if you take nasal vaccine and you develop local IG antibodies to that specific virus. Now, for how long? See, these are going to stay for a few weeks, maybe 4, 5, 6 weeks, not beyond that. So are we asking about getting a vaccine, nasal vaccine, which we take every 6 weeks? No, this is not a practical answer. So community transmission is not, I think we should not be asking for it. And eventually the new strains which will be coming in future, either in an endemic form or a pandemic form, we don't know, but Omicron is a good example. It came like anything and it infected majority of the population. Nobody was spared, and community transmission is very high. R0 was in 20s at some places. So community transmission cannot be stopped with vaccination. It is too far-fetched a thought.
Simple answer is that vaccines will not stop the community transmission. So we should not hope for that. Okay. Gaurav, how important are boosters? Because the primary vaccination, most of, at least all of Delhi's had the primary series, at least one dose everybody's had. So how important are boosters? Now the talk is about boosters, precautionary dose. So I'll ask you all these questions. How important are boosters? Why a so-called precautionary dose? And what is the rationale that Government of India is following a 9-month gap for the third dose?
I think that's the question on top of everybody's mind. Do I need a booster? So what WHO says about boosters is that, is there a need? There are three things to consider. Is for people who did not respond to the primary immunization schedule of the 2 doses, so those who were immunocompromised, so to say, if there is significant waning of immunity, then boosters would be required. And the third would be to protect against newer variants. So for the first category, definitely boosters are required, the immune compromised or those who did not respond to the first primary course. For the waning of immunity, as Vipul also showed, that there is still good immunity against severe disease and hospitalization and death provided by the primary vaccination schedule of 2 doses. And WHO is continuing to say the same. As far as the protection against variants, I think in the next few slides we'll see. So for India, there are only anecdotal reports like this one from Mumbai, where they said 96% of the deaths are, or the oxygen requiring patients are, those who were unvaccinated. And Delhi health minister said 75% deaths are in unvaccinated, although that's not very reassuring to me, because 25% deaths, if he's saying, are in vaccinated. But still, if we look at hard numbers, they are not available from our country, which is extremely unfortunate, that 2 years of so many cases and we've not been able to generate good data. But we look towards US as usual, and this chart sums it up. The unvaccinated, the number of deaths is around 10 per 100,000 as compared to the fully vaccinated primary schedule. This is not for the boosted, but the primary schedule, and that's almost near zero. But then there are studies which have shown that the effectiveness of the vaccine against symptomatic disease, not the death or hospitalization, but overall symptomatic disease, goes down. And by 6 months, the data shows that more than 30% of the recipients, the vaccine effectiveness significantly wanes. And once given a booster, that goes up again to above 90%. So this was in the Delta wave that this was studied. Next, please. And this Vipul has already shown. Again, 6 months is the time where most studies are showing that there is some waning of immunity against Delta, not very significant. But for Omicron, definitely the vaccine effectiveness, which as it is, isn't as good as against Delta, but it falls significantly at 6 months. But if a third dose is given, this is again all US data, all data for mRNA vaccines. So not directly extrapolatable to us, but still, this is what we have. And there's a very recent report from 23rd January from a group of hospitals across the US, which have shown that only 24% of all COVID hospitalizations are in the fully vaccinated, and only 8% of those hospitalized are boosted. And in the ICUs, the numbers are more encouraging, that only 14% in the ICUs, patients in the ICUs are fully vaccinated. These include the very elderly, the ones with the comorbidities. So definitely, even people who got 2 doses, they are getting protection. And I think this is the slide or the data which is being talked about the most all across the world. There are similar data available from other developed countries also, which show that as compared to the unvaccinated, which US is unfortunate enough to have a lot, more than 20% of their population is refusing vaccination. So as compared to the unvaccinated, the boosted are 68-fold protected against death. That is 99% protection is being provided by 3 doses. Those who did not get a booster, even they are getting more than 95% protection against death. But still, giving a booster in the US is reducing the death chances of death 7-fold. And there is data from both the mRNA manufacturers. The panel above, panel A, is from Pfizer, and the panel B, the one below, is from Moderna. And again, both of them show that in the unboosted, the incidence of having a symptomatic infection, that might just be a mild URI, but a symptomatic infection is about 5 to 6%. Whereas those who have received a booster or third dose, the incidence of symptomatic infection goes down to about 2%. So there is some protection from the booster. Now looking at UK as well as US data, and again, this shows that for Omicron, this is for Omicron, that for 2 doses, the vaccine effectiveness wanes, but once a booster is given, it goes up around 90%. The next slide, please. So why the basic premise which we need to understand, as Vipul also explained, that for infections, the symptomatic infections, the principal defense is the neutralizing antibodies, mostly what are available in the upper airways, and 2 shots are giving about 30% protection against a symptomatic infection against Omicron. Whereas severe disease, hospitalization and death, 2 shots is giving about 50%. And this is all data from UK and mostly from the developed world, I should say. So 3 doses takes the protection to 60% against symptomatic infection and around 80 to 90% after 2 to 3 months of a booster. Next slide, please. Now, why the 2 doses are still holding up to give us good protection against severe disease and hospitalization is because of the preservation of T cell reactivity. That's the bottom right corner of this image, if you see, and the bottom left corner of this image, if you see. So the B cell reactivity falls against Omicron from at 6 months to about 42%, whereas the T cell reactivity, that's the bottom right corner, remains about 85 to 90%, thereby giving us good protection against severe disease and death. And it's been rightly pointed out by a number of studies that it's not that Omicron is mild, it's that it is encountering a population which is vaccinated or previously exposed. It is just about 27-30% milder than Delta, but because the population is either protected due to prior natural infection or vaccination, the results are better. Now, if we do think of boosters, how many, how frequently, and Israel has gone ahead and now given approval or go ahead yesterday, I think, to give the 4th dose to all adults. And they say that getting the 4th dose gives a 3-fold protection even against severe disease, although no data has yet been shared. Now, next slide, please. Now, if we look at our own situation, we do not have any, let's say, real life data to say what our population should be doing, especially since we are not using mRNA vaccines, we are using the AstraZeneca platform, the viral vector, or the inactivated Covaxin. So there's this paper, the COV boost study, where they followed up an AstraZeneca primary schedule by 6 different vaccines as boosters. And this is all that we have to kind of base our decisions on. Next slide, sir. So if with a primary series of Covishield, a homologous booster or the same Covishield is used as a booster, we would expect a 3-fold rise in the antibodies which are basically responsible for the immediate protection against symptomatic infection or asymptomatic infection. But there is no boost in the cellular response, which is responsible for the long-term immunity against the severe disease and hospitalization. But if a heterologous booster was used, mRNA vaccine gave the best boost to the antibodies. That's about 20 to 25 fold. But unfortunately, we do not have access to that. Now, Novavax, or the upcoming, which is now going to be launched in India's Covovax by Serum, gave an 8-fold rise in antibodies when used as a booster after a primary series of Covishield. And it also gave a 3 to 4-fold boost in cellular immunity, even better than the mRNA vaccine. This study used one inactivated vaccine, which is not in use anymore, but that we could take that as a surrogate marker for Covaxin. And it did show some rise in antibodies there, but there was no boost in cellular immunity. So when we talk about Indian population, those who have received Covishield, they would expect a very, very modest boost in the antibody response when they use another Covishield dose as a booster. But for Covaxin primary schedule, we do not have any precedent or any real world data or even good quality lab data. The previous study that I just showed again is basically a lab data for the antibodies that would be produced, but not real life effectiveness data as we have from the US. So if and when we should use a booster, I think now what most practical experts are suggesting is that since Omicron is already on the way out, at least in Delhi, whenever we are expecting or anticipating another wave, looking at the West, before that is the time that we should be going for a booster maybe. And if we have access to Covovax soon, which hopefully we would, that would be a better booster than taking Covishield. Those who received Covaxin as the primary schedule maybe should be going for Covishield as a booster. Now, why India is calling it a precautionary dose? Well, we are like that only. We are different. So in the whole world, nobody else uses this term. Maybe the reason could be that booster is a more scientific term and you need more evidence, more studies to say that it's going to be a good booster. And this is the same vaccine being used, and there's no hard evidence that it is going to be a really good booster. So it's a precautionary dose. And for the Vipul wants to add something.
Vipul wants to come in. Yeah, Vipul, go ahead.
You're
Mute.
Yeah, sorry. I think somebody wisely advised them to use precautionary word over here, because see, for 83% of us got Covishield, and Covishield by design is a single dose vaccine. Once you consume the adenovirus, you cannot take the same adenovirus for another. You cannot use the same adenovirus as a vector again repeatedly. So smartly, Russians used the second adenovirus for the second dose because they knew the same adenovirus cannot be used again.
Different platform was used for the second dose.
And Johnson & Johnson introduced as a single dose because they knew that using it as a second dose will not be very wise. But Covishield, I don't know why, just to increase 3-4% of their immunogenicity, I should say, they added the second dose also with the same adenovirus. So taking third dose with the same adenovirus is actually just a precautionary dose. It is not going to add anything. Just दिल की तसल्ली देगा वो. Marginal antibody अगर वो increase हो भी जाएंगी, वो I think they will be at the cost of circulating antigen presenting cells, which might be needed in future for actual infection. Rather, I am very skeptical about using third dose of adenovirus vector vaccine. This can be counterproductive also.
Yeah. So we had this discussion, Vipul and me, recently, that when Covishield was released, I think there was talk that this is not amenable for boost because our own immunity would against the chimpanzee adenovirus would not let it have the desired result. Coming to the last part of the question, the gap, again, this is something different from the rest of the world because US is using a 5-month gap from the second dose. UK is saying 3 months gap from the second, from the primary schedule. Why we went for 9 months? Again, there is no, so we are very secretive when we make policies. We don't kind of open up why we are making policies in our country. But basically, there is some data which says that 6 to 9 months after AstraZeneca vaccine is the right time to give a booster, and we chose 9 months. Most of the people, the healthcare professionals and the elderlies with comorbidities would have completed 9 months by now from the second dose, so they would be eligible for it, and not the general population. But 6 months is what most of the evidence says is when the immunity starts waning. But as I said, most of the evidence is on mRNA vaccines from the West, and there's hardly any data from India.
Puneet wants
Maybe
to
one
comment.
reason is to reduce the number of eligibles.
Yeah,
If it
that's what I
was
said.
4 months, 6 months, then there will be more rush at the vaccination center. So that might be the thinking behind the process. Although it is
Yes,
not,
true.
it is not science, it is logistics. You can.
Yes.
So if we, for the, at the risk of sticking my neck out, do I understand that people who had Covaxin, they can go ahead and get the third dose, precautionary dose, and it wouldn't harm them at least? As far as the Covishield population goes, it's probably at this moment best not to take the Covishield booster or precautionary dose.
I think that
I
would be
don't
going
think we
against
can
the.
recommend that, but that is what most of us would be thinking of. But Covaxin
You know,
primary schedule ones should take Covishield as a booster and not Covaxin. Vipul wanted to say something.
Yeah, I think going against the government policy would be tough at this point of time. Whosoever wish that antibodies should be there as far at a good pace as soon as possible, they should go for the precautionary dose, whatsoever primary series was, because Covovax is coming. God knows when is it coming. 10 days, 10 months. Government doesn't. In India, you never know what will be the pace of delivery. So I think if you are if you think that you need a third dose, you should go. And if you are confident that your first 2 doses have worked well, you can wait.
Okay. So it's pretty inconclusive at the moment. Fair
Actually,
enough.
it is.
Okay, so I think we've had fair amount of data already presented on this homologous, heterologous, but Puneet, once again, I would request you to quickly go through this. That earlier times we used to follow the same vaccine, same brand as a booster or as a second dose, but now the talk is about this heterologous. So why? What's the
There are 2, 3
science behind
reasons.
it?
One, we need very high supply of vaccine, which may be not possible. Often on we have seen shortages. So one reason to try was that only, to tackle the shortages. And the vaccines are also new. The virus is new. If some new adverse effect comes against one vaccine, like TTS came with viral vector vaccine. So if we have a possibility of heterologous, completing the schedule with heterologous thing, so the schedule can be completed, the population can be immunized. Secondly, as the vaccine efficacy is decreasing with one or the other variant, then one thought process is to find something better with the same vaccine. So the thought is, if different vaccines are given for the same virus on a different platform, the breadth of the immune response is better, maybe better. And many studies have really shown that it is really better. So I think it has started with COVID vaccines, and this may be the process for other vaccines also, other viruses also. That in giving the different vaccines for the same virus increases the breadth of the response. The heterologous schedules, it is either primary heterologous schedule and one is heterologous booster. In heterologous primary schedule, the first 2 doses are of different platform. That is called prime boost strategy. As Dr. Vipul Jain said, this Sputnik vaccine itself is kind of heterologous, like two different carriers have been used for 2 different doses. but in other ways to give vaccine based on different platform itself. In Sputnik, the platform is the same, viral vector. In Prime Boost, the technology is different for the both doses. This has been tried. And the trial started very early in 2021 when the deployment was being started. Some researchers started this testing these heterologous schedules. The UK group started this. They started testing combination of Oxford vaccine and Pfizer vaccine, given first Oxford, then Pfizer, and reverse at 4-week interval, then 12-week interval, and they found that the immunogenicity is better when the two different vaccines are used as the first and second doses, though at the cost of increased reactogenicity. Reactogenicity in the form of like local symptoms, fever, body aches, etc. Those are little higher when heterologous schedule is used. This is for primary schedule. Next slide, please. So they when they tested, when the first first they started with Oxford and Pfizer vaccine, then Moderna and Novavax were also added, and different combinations were studied. When the Oxford vaccine was given, Oxford is also Oxford AstraZeneca, that is Covishield vaccine, was given as the first dose, and the different vaccines were used as a booster. When the same homologous second dose was given, the titer was 1971. When Moderna was given, see how the titer went up. It is 20,000, many times than the homologous. This is when the second dose is different. And when Novavax was used, again the titers was many times, like at least 3 times the homologous. So similarly with Pfizer vaccine, when Pfizer vaccine was used as the first dose and the different vaccines were tried as a second dose, with homologous it was 16,000, with Moderna it was a little bit more. But Novavax, with when first dose of Pfizer and second dose of Novavax, the titers were even lesser. Although they were significant, but they were lesser than homologous. That is why the evidence for heterologous schedule is better when the viral vector vaccine is the first dose. After mRNA vaccine, the evidence is less there. So, and these are the initial efficacy studies. Now, vaccine studies are also available, real world effectiveness studies. In Sweden, they have compared 94,000 individuals who took Oxford or Pfizer vaccine, Oxford vaccine as first dose and Pfizer as second dose, compared to 16,000 individuals who took Oxford as the first dose, Moderna vaccine as the second dose, and 4 lakh individuals who were homologous to Oxford vaccine. And this was also compared to so many unvaccinated individuals. And on follow-up, it was found that the heterologous combinations are much better. If in the first combination, it was 79% efficacy, in the second combination was 68%, and with homologous, it was just 50%. So there was a significant difference in the heterologous schedule. Now comes the heterologous booster, like when the first 2 doses are of the same vaccine and you boost with a different booster. The first study was from US. It is available as a preprint as of now. They studied adults who received one of the 3 vaccines. Their 3 vaccines were available: Pfizer vaccine, Moderna mRNA vaccine, and Janssen vaccine. Janssen is a viral vector vaccine, single dose vaccine. It was given and booster of any of the 3. So they studied the 9 combinations when the first 3 vaccines were either 3 and followed by booster of either 3. And this also found that when booster is given, the increasing neutralization activity against the virus is 4.2 to 76 folds for all combinations. Homologous boost was less. When heterologous booster is given, the titers are much higher. And titers were highest when Oxford vaccine was followed by Pfizer or Moderna vaccine. Reactogenicity in this study was similar. And next was the Comcov study from the UK. They have also studied the same study which was shared by Dr. Gaurav Sethi also, when the 7 different vaccines they have tried as a booster, when the priming has been done by either Oxford vaccine or the Pfizer vaccine. Again, it was shown that the spike antigen, the titers, the ratio of the titers was much more when the heterologous booster is given. Next slide. And same with whether the primary doses of is Pfizer vaccine or of the viral vector vaccine, when a booster is given of a different vaccine, the titers are much, much higher. Again, as Dr. Gaurav Sethi said, when the primary vaccine is of Oxford vaccine, the cellular boost is not there with the homologous one. If we give third dose of Oxford vaccine only after 2 doses of Oxford vaccine, the cellular boost response is not boosted. Only antibody titers are increased. This is better with if the 2 doses of Oxford vaccine are followed by Novavax, that is subunit vaccine, then the cellular response is even better than the Pfizer vaccine. This cohort is still being followed up currently. And vaccine effectiveness for the heterologous booster is also available. This has also shown that symptomatic disease is compared by 93% to unvaccinated individuals, whereas the efficacy is 87% who had received the same dose. So heterologous booster is better. Same effectiveness studies from Chile also, it has also shown when CoronaVac, that is inactivated vaccine, so we can compare with with our Covaxin. It was followed by a booster of CoronaVac or Pfizer or Oxford vaccine. When the homologous booster was there, the efficacy was 68% against infection and 71% against symptomatic disease. If this inactivated vaccine was followed by the viral vector vaccine, Oxford vaccine, the efficacy was 90% against infection and 93% against symptomatic disease. And if the booster was of Pfizer, then again the effectiveness was much better. So seeing all these data, the current recommendations of EMA and ECDC, European Center for Disease Control, is that heterologous should be preferred if considering the pending the further infra data, otherwise it is preferable. Data is stronger when the viral vector vaccine is the first vaccine, followed by a booster with mRNA. The data is less for mRNA vaccine as a primary, followed by a viral vector as the reverse. And since all these studies are smaller, the risk of myocarditis is yet to be found out. As we know, with Moderna vaccine, there is some risk of myocarditis. The risk for this heterologous schedule is yet to be determined. And obviously, the data is less for special situations like immunosuppressant disease. WHO also says, although the current standard of care is homologous schedule only, the first and foremost target is high coverage with any schedule, depending upon the availability of the vaccine. First 2 doses of any schedule is much more important than the suitability of schedules. Otherwise, countries implementing the inactivated vaccine should consider viral vector vaccine or mRNA vaccine as a booster. Those implementing vector vaccine may consider mRNA for a booster, and those implementing as mRNA vaccine may consider as a viral vector vaccine, although the evidence for the last is little less.
Okay, so most of the recommendations are for heterologous boosters, while we still are not on the same page as far as Government of India goes. Anyway, time will tell. Sir, now हमारे काम का तो vaccinating kids है. Of course, 1 साल के बच्चे की भी माँ पूछती है कि वैक्सीन कब लगेगा, 5 साल की भी, 12 साल की भी. Of course, we know for us it's only 15 to 18. So what's the status as far as the recommendations go? Government of India, of course, we know, but WHO and other countries, how are they vaccinating their children?
Well, all the discussion that we've done till now is mainly based on adult data. And very few countries, well, several countries have started vaccinating children. Data is still being collected. So let's see what WHO says about it. Now, WHO stand, as I could get on the 20, they have updated on 12th of January this year. There's a strategic advisory group of experts has concluded that Pfizer vaccine is so safe to be used for those who are aged 5 years and above, and Moderna more than 12 years. This is what WHO is saying. The companies may be saying something different. A smaller dosage is required for children than for adults. Now, children and adolescents at high risk of severe COVID may be offered the vaccine along with other priority groups for vaccination. Children and adolescents tend to have milder disease compared to adults. So, unless they are part of a group at higher risk of severe COVID, the priority should be to fully vaccinate the world. So, WHO says only selected high-risk children should be vaccinated and not all children, unlike many countries, including ours now. Now, some countries have given emergency use authorization for mRNA vaccine for use in adolescent age group 12 to 17, which has been developed by Pfizer and Moderna. In November 21, one stringent regulatory authority approved mRNA for 5 to 11 years also. Probably they're meaning FDA. trials in children as young as 3 years were completed for 2 inactivated vaccines, Sinovac, Coronavac vaccine. And then these products were approved by Chinese authorities for the age 3 to 17 years. Although WHO says they have not approved this for children, although this is approved for adults. Now our own, next one please. Now Indian vaccine, Covaxin is an adjuvant inactivated vaccine which has been developed by Bharat, we know, was approved for in India for the age 12 to 17 years recently. Now, the Indian regulatory authorities have given approval to Zycov-D also, which has not yet hit the market. And several COVID-19 vaccines are undergoing trials in younger age group in various parts of the country, various parts of the world. So everybody is moving downwards. In fact, there's a trial going on by Pfizer where they use 3 doses. 2 doses had not shown very good response in 2 to 5 years. So they have changed the strategy, and now they're giving 3 doses as the primary vaccination schedule for children less than 5 years of age. In fact, they have included 6 months to 2 years age group also in that. Probably they're smelling some success, otherwise they won't have included younger children also. So probably 3 doses is what will come for younger children. Of course, it's a smaller dose that is to be used for children, whether 1/3 or less, that depends on vaccine to vaccine.
Sorry, I muted myself. Sir, are we stuck at 15 to 18 just because of the logistics and we don't have that number of vaccines, or is there any other reason? I mean, why not 12 plus?
Well, I think, of course, again, as has been discussed, we don't know the exact reasons in the government of India, and their special people do not reveal that. But it seems the availability is also a factor. Of course, in this age group, it's a priority compared to younger children. They're suppose 10th class and 12th class, and their need in that sense is more. MISC is also somewhat more in older children. So in those taking those factors, yes, that was a good idea. And then we don't want to overwhelm our system of even vaccinating and creating a panic all around. So probably it's okay to start with 15 and 18. And now that we have covered a fair number of this age group, we can move downwards.
So, Dr. Puneet, I'm sure आपके क्लिनिक में भी पेशेंट आते होंगे, पूछते होंगे कि डॉक्टर साहब बच्चों को कोविड होता भी है. I think the first question they ask, and then you have to tell them, हाँ भैया कोविड होता है. तो फिर the next question is that फिर वैक्सीन भी लगना चाहिए, and you have to tell them कि अभी वैक्सीन है नहीं. तो but our sense, what we've seen over the past 2 years, is that children definitely have a milder disease compared to adults most of the times. Of course, a few of them are unlucky and land up in hospital, but most of it has been missy rather than the acute illness as such. So given that in children it behaves like a normal viral fever or slightly a more severe form of flu, but they get away fine, do the children need the vaccine?
Again, among so many unique things, this is also unique about COVID. For all vaccines, we usually think about childhood infections. Vaccines are always developed for childhood infections, and then they move upwards. It is reverse. Since the mortality and morbidity is maximum in the highest age group, so all these vaccines have been started in that age group, and then we are moving downwards. Obviously we have seen that children have only mild disease. That is why they are at the least priority. But isn't natural immunity better? That may not be there, because we still do not know about the long-term effects, as Dr. Vipul Jain also explained nicely, that it can affect T-cell immunity. Children are still developing immunity. It can lead to increased autoimmune disorders. So it is not an idea to just leave it for natural immunity. Vaccine is required, but the priority is higher age groups first, and then move downwards. And by that time, we will have more stronger data, and we are also hoping for better vaccines also, second generation COVID vaccines. So it is a good idea to start with 15 to 18 only, and then move downwards.
Then again,
And
the
regarding-
great Indian puzzle that when we have both Covishield and Covaxin, why only Covaxin for children, and where is the studies or the
Covishield.
data for
Actually-
Covaxin? Nothing in the public domain, at least.
We usually AF the West, whatever West does. The research started on Covishield also on adolescents. Incidentally, when the trial started, at the same time, this new adverse effect of TTS in adults, young adults came up, which is the thrombosis with thrombocytopenia syndrome. Since those cases reported, the trial was halted. It was actually paused, and it was said that it will be resumed after the complete investigation of the TTS. Although TTS was found to be very rare, the trial never resumed. I don't know why. The trial was not resumed, so we don't have any data. Since there's no data and we are not doing anything, so we say we are not going Covishield. Although Russians are giving the same vaccine, they are giving the Sputnik vaccine to their adolescents. It has been recently started in 12 to 17 age group. They are reducing using the reduced dose, Sputnik 5. 1/5 of that dose, they are calling it Sputnik M, and they have started vaccinating the adolescents. Russians are doing very differently. Sputnik V is vaccine for adults. Sputnik M is vaccine for same vaccine. 1/5 dose is for kids. First dose of the Sputnik is Sputnik Light. They have licensed it separately. It is Sputnik Light. They have even tried Sputnik, the same vaccine as the nasal vaccine also. Same vaccine they are trying on as a nasal vaccine. So viral vector vaccine is being used. In fact, the Bharat Biotech, which is developing a nasal vaccine in India, that is also a viral vector vaccine. So viral vector vaccine is also being tried in kids. And there are high hopes. It is said to be a game changer because it can induce the local mucosal immunity, which can affect the transmission also. So it should be tried, in my opinion, although there is no trial, so it is not started for Covishield.
Okay, so at the moment, Covaxin 15 to 18 years of age.
Right. And regarding safety data, only small data is there. For Covaxin, they recruited 976 volunteers in 2 to 18 age group, and out of which 342 were found to be seropositive at the baseline. So they were excluded. So only 526 they could do. The 526 were further divided into 3 age groups: 2 to 6, 6 to 12, and 12 to 18. Whatever data, interim data is available, the vaccine was well tolerated and with the similar reactogenicity profile as with older age groups. So that is all we have as of now.
Okay, now Dr. Vipul, coming to more day-to-day conundrums that we face. So how soon can a child get the routine vaccine shots after a confirmed COVID, which probably is a more straightforward answer, a likely COVID, because nowadays we hear these stories कि डॉक्टर साहब मेरे को हुआ था, बच्चे को भी symptoms थे, उसको तो हमने टेस्ट कराया नहीं, but likely COVID ही होगा. So how do we advise these parents for their routine vaccines? One after an RT-PCR confirmed COVID and 2 after a likely COVID. And does the severity of the illness, because children have very mild illness, 2 दिन fever हुआ, तीसरे दिन ठीक हो गए. So
there is no answer found anywhere in the literature regarding this, but Government of India has laid down guidelines that if you are a confirmed COVID case, RT-PCR positive, you have to wait 90 days post-recovery to take the vaccine. That is one guideline. And
but that is for COVID, the routine vaccine का.
routine vaccines.
PPT लगना है उसका 1.5 साल का, MMR लगना है.
Sorry, routine vaccine shots. I'm really
Yeah.
sorry.
I mean, if
No,
the
no, we
child was
don't
due
do-
for his MMR or chicken pox.
Okay.
now he's
No.
dead.
Sorry, I'm really sorry. I thought we are talking about
No,
COVID.
not the
vaccine.
COVID vaccine, not the COVID vaccine. Routine vaccines.
In either case, likely COVID or confirmed COVID, as the patient recovers, we should restart the routine vaccination as soon as possible. Child is ready to take the vaccine. There is nothing, no gap required. And as far as severity of the illness is concerned, if the child has received blood products during hospital admission, then we need to wait for 4 weeks for live vaccines. Otherwise, all vaccines can be given as the child recovers.
So this I guess we have a very clear answer that as soon as the child recovers from his COVID illness, he can go ahead with his routine vaccination as soon as he is clinically recovered. Only if there are blood products or a prolonged course of steroids, then the usual gap of at least 4 to 6 weeks should be there. Is that clear?
Yeah, I think most of the kids, yes, in case they need long course of steroid, which is not usually needed, pulse is given for 4 or 5 days, then you don't need to wait for any duration to give any vaccine.
Okay, fair enough. And Dr. Gaurav, how soon can I get my precautionary dose? So now we are talking about the COVID vaccine. After
that's why I interrupted Vipul. मेरे सवाल का जवाब वो दे रहा था.
Encroaching on your territory.
But he's allowed to do that. No, so once I get a confirmed COVID or a likely COVID, which many of us have got in this Omicron wave, when to get the precautionary dose, so to say. So if we look at the West again, as per the US CDC, as soon as you recover from a COVID infection, you should just go ahead and take your booster, or if you're in the primary schedule, still you take your vaccine as and when you recover. There is no gap or no waiting period advised by them, unless and until you were given monoclonal antibodies or plasma. Then they advise that you wait for 3 months, 90 days to take your COVID vaccine after suffering a COVID. If you look at UK NHS, they say that once you get infected with COVID, you wait for 4 weeks before taking a COVID vaccine. Now, in the second wave, April, May 2021, when many healthcare professionals were seriously ill, or many people were seriously ill, the guidelines came out from the Indian government that if you get a COVID infection, you should wait for 3 months before taking a COVID vaccine. As of now, I think that holds for India, but since many people are not getting PCRs done, and PCRs are not linked to and the reports don't get linked to the Aadhaar, so there's nobody stopping you from taking a COVID vaccine. But so the short answer is that if you look at the West, you don't need to wait once you've recovered, unless you've received monoclonal antibodies. And if you continue to follow what the government of India recommends, you wait for 3 months. But as I said earlier, also many experts who are dealing with COVID these days say that अब ये Omicron wave तो almost निपट ही गया. Now you get your boosters once the next wave is upcoming, or as Vipul said, if you trust your immunity and you feel you've developed a good immune response, you just hold on with your boosters. And if you feel नहीं यार मेरे को तो लगवाना चाहिए, so you go ahead with the COVID vaccine once you recover.
So precautionary dose can be taken immediately after you've recovered. Can be, yes, if you want to.
Yeah.
Fair enough. So this is a famous no-vaxxer who's come into the limelight, and we have a few patients, at least their parents rather, I would say. And though the government of Delhi says that all our adult population has received 1 dose at least, I at least for one have parents who come and tell me, Doctor, मैंने तो लगवाया नहीं है. So and they tell us कि सर मेरे को तो अप्रैल 2020 में भी हो गया था, अभी भी ओमिक्रॉन हो गया, 2 बार तो कोविड हो चुका है, तो अब तो नेचुरल इम्यूनिटी होगी, तो अब क्या करना है वैक्सीन? तो डॉक्टर विपुल, योर आंसर टू दैट?
Partially, we have answered this question before. Next, please. If you look at this, we are falsely being caught in antibody discussion, which is going on social media. कि antibodies कितनी है? वही वाली बात है, कितनी देती है वाला वैक्सीन के साथ हो गया है, in comparison to what we would be looking at. How many memory cells have been created? Nobody is able to see that. So everyone has been after antibodies कितनी है, और ये सब के चक्कर में हम पड़ गए हैं. So antibodies fall with time, they go away. And your natural infection, actually, you don't know how much it got into your body and how much memory cells it has produced. Vaccine for sure is, since it is being injected inside the body, gives good amount of memory cells for a good amount of time. It is difficult to measure as such how much memory you have, but definitely over a time it has been seen that in long run it is the T cell, B cell memory which is going to save you from hospitalization, from oxygenation, from ICU admission. And it is not just the antibodies which what you measure is going to save you. So the second question which was there, that how many antibodies, how much antibodies, how many antibodies I have, from where I have to get it measured. It does not arise. There is no standardization of any kind of antibodies, whether they are spike protein antibodies or neutralizing antibodies. They are only for research purpose. They are only for they are only research tools for publishing data, but practically they have no role. And in a previous question, I have answered that the initial defense of B memory cells are good enough to save you from Delta once you have taken vaccine for Wuhan strain. Now, this also we discussed that 80% of the epitopes in Omicron were same as in Wuhan strain. So which is the most mutated strain at this point of time? Almost 50 mutations which are known in it. Now, very interesting about Omicron that happened is, a lot of people say why the lung is not involved in Omicron. Two major mutations, basically, they affect the entry of the Omicron into the cell. That is why it is not being able to enter into the cell. It is not being able to affect the lung the way Wuhan or Delta was affecting. That is the primary reason. I don't know whether this vaccine status is being the reason of less virulence of Omicron or whatsoever. But the message here is strains are going to come in future. Natural infection doesn't give you good amount of memory in terms of reservoir, and so taking vaccine post natural infection is important.
Okay, so clear message that even though you've had the infection, but please still go ahead and get your vaccine. Sir, any safety concerns around the COVID-19 vaccination? And do we have data from India, or again, it's nebulous and hidden?
Well, it is a very important point that any vaccine that comes, including, of course, this COVID, we should know what are the side effects, how frequently they're occurring, and when they occur, how are the people recovering out of it. That kind of data is available from US in good numbers and all. What if you search for India, this is what you'll get. You can pain, redness, and swelling, and so on. There's nothing very serious, as if it's something that the public doesn't want to know. That's how it seems government of India wants to take this issue. Next slide, please. But major complications are anaphylaxis, which we know is very rare. It has occurred in approximately 5 people per million vaccinated in the US. Next, the second important one is this TTS, the thrombocytopenia syndrome. Third is myocarditis and pericarditis. Fourth, Guillain-Barré syndrome, and deaths, whether because of these 4 or some unknown associations. Now, if we take TTS, as the US data says, that it is a rare but serious adverse event that causes blood clots in large blood vessels and is accompanied with low platelets. Now, this is a kind of paradox. Platelets are low, blood clotting is occurring in various vessels. As of January 13, 2022, more than 17.8 million doses of J&J vaccine had been given in the US. And out of that, 57 confirmed cases of TTS was seen. Now, this is JJS vaccine seems to be causing it more frequently. And the next one, and to date, according to US data, there are just 3 confirmed cases of TTS following the mRNA vaccines of Moderna. Now, if you look at the NHS data about the same syndrome, although this condition remains extremely rare, they say, there is a higher risk in people after the first dose of AZ vaccine, the same vaccine that we are using as Covishield. To date and overall, just over 10 people developed this condition for over 1 million doses of AZ vaccine. Now, this is seen more often in younger people and tends to occur between 4 days and 4 weeks after the vaccination. So what they have done is they have changed the strategy a little bit, that those who are 18 to 40 years age in the UK, they're saying avoid the AZ vaccine. But if the first dose has been taken and you did not develop any problems, second dose can be taken because this side effects tends to come mainly with the first dose. Next, please. Now about Guillain-Barré syndrome, after more than 17.8 million doses, there are 301 preliminary reports of GBS. These cases have largely been reported about 2 weeks after vaccination and mainly in men, many in those who are 50 years and older. So unlike the TTS, this is occurring in older people. Based on the data, the rate of GBS within the first 21 days, it is 21 times higher than the Moderna or the Pfizer vaccine with JJ vaccine. After the first 42 days, the rate of GBS was 11 times higher following the JJS vaccine, and analysis found no increases with Pfizer or Moderna. So it is again occurring mainly with JJS vaccine. Now, myocarditis and pericarditis, here the reverse thing is happening. As of January 13, 2103 preliminary reports of myocarditis or pericarditis were reported to the CDC, and mainly occurring in 30 years and younger. Most cases have been reported with Pfizer and Moderna vaccines. Though follow-up, including medical record reviews, says that CDC has verified 1,213 reports. So fairly large number of myocarditis people, side effects have been seen. They have been seen, unlike the other ones with Moderna vaccine and the Pfizer vaccine, they have occurred in younger age group and not in the older age group. So these are somewhat different, but all of them have recovered easily with proper treatment and some without treatment. Now, deaths, more than 529 million doses of COVID-19 vaccine were administered in the US from December 14, 2020 to January 18, 22. During this time, they received 11,468 cases of reports of death among people who had received this. And continuing monitoring has been identified, has confirmed that there are 9 deaths at least with JJ vaccine. So confirmed deaths are very few, but associations suspected have been 11,000, which is still not a very large number considering that such huge numbers have been vaccinated in that country. Now coming to our own tragedy. Data is just not there. I'll show you where this data came from. There's a commentary in BMJ published 4 days back in which this has been, this is the one where it has been kind of, it's a very indicting report on the scene in India. It says that people are not being informed, and then even the families are not being informed of those who have died. Even they don't know why that person died, although clinically it's very clear, but government of India is not confirming to them. They have quoted a few cases here and there. Let's go back to the other. Okay, so now they say that 30th November 2021, the National Committee had received 49,819 adverse event reports, almost 50,000. By then, India had administered 1.23 billion doses. This says that there are just 4 adverse events per 1 lakh unit doses. This is not acceptable. If less than 10 are being reported, your reporting system is in doubt. This is what the international standards say. In contrast, the Canadian safety surveillance system has reported 48 per lakh, and in UK it has been 300 to 700 per lakh. We are just 4. Obviously, we are not reporting. Next, please. Now, reports for TTS are similarly low as overall. Only 26 reports have reached the national committee so far. And the reports that do reach the national committee are investigated slowly. Of this 946 cases reported up to November 21, committee had completed investigation for only 89 till very recently. And of the 26 TTS cases by mid-December 21, only 6 have been investigated at the time of writing, with 5 attributed to Covishield. Now, this is how slow or, okay, whatever you want to say, the system that is there for safety surveillance in our country is that once a hospital or a healthcare provider reports, then it goes to a district committee, then to a state committee, then to a national committee, and then the national committee will verify, and then it is supposed to close the loop by informing the people who are injecting, who are vaccinating, and the person who has suffered. But obviously the loop has not yet been closed even in a single case. Next, please. Now, hospitals are failing to report adverse events following immunization, AEFIs, while patients and their families don't know how to report it. There is a system. The system is also constrained by other problems, including the slow pace at which officials are investigating whether reported adverse events are due to vaccine, and the non-communication of these findings to the patients and to the vaccinators. So we have problems, but it is quite clear that vaccine is not 100% safe, just like any other vaccine. It is not more unsafe or causing too many adverse events, at least not the severe and the very, let's say the ones which will end up a patient in the hospital or in the ICU or deaths. Those are very few, but yes, they are there.
Chalta hai attitude is percolating everywhere. It is unfortunate because itne large scale pe to koi vaccine or India mein laga nahi.
Yes.
And if the CoWIN app can track our vaccination schedules so well, it could very well have tracked the adverse effects also, and we would have had data much more than rest of the world combined, probably, given the number of doses we've given. It's a very sad state of affairs, and somehow it kind of, at least for the healthcare professional, it brings down the confidence in the whole process, the vaccine and the process of vaccination.
Indeed, we have lost opportunity, actually. We would have taught the world what it can cause.
Because CoWIN app, we have a wonderful app actually to deliver the vaccine. Why not track the rest of it? Anyway, that's the way India works. So, Dr. Puneet, given all the variants which are going to come, I think we've kind of partially already answered this question. Puneet, are you there or have you lost connection?
Just a minute. Yeah.
Okay. So there are reports and I think rather comments from the vaccine manufacturers themselves कि ये तो अब हर साल vaccine लगाना पड़ेगा. So are there going to be repeated boosters, annual shots like the flu vaccine in the future?
First of all, even annual shots are not sufficient for flu itself. It is for the West where there was only one season. Anyway, yes. Secondly, we have to see why we have to give booster. What is the purpose of the booster? If the purpose is to prevent infection, then if we are using the current vaccines, we are following a mirage. It has been shown that if we give booster, the titers are increased for a few weeks, and then it wanes. It has to wane. The half-life of IgG is just 25 days. So the titers we can only transiently increase, and it can only prevent infection for a short time. After that, it will wane off, as Dr. Vipul Jain also explained nicely, that for long-term effect, even 2 doses are sufficient. They are giving enough memory cells, T cells to give reasonable protection against the severe disease for most of the strains. And if there's further change, we may need even better vaccines, not annual boosters. We need better vaccines. And another issue is obviously the vaccine equity. Each dose we are giving as a 3rd dose or a 4th dose as has been started in some countries. The primary dose of someone is being delayed. As of now, only the population coverage with the full primary series is less than 10% in 45 countries and less than 40% in 105 countries. We have reached 65%, but there are so many countries who have not. And in that case, the transmission will go on. There will be more and more mutants, and it will again, we will be in vicious circle. So the first priority should be first 2 doses as soon as possible to the whole population rather than the annual boosters. In US, they also studied the direct effect of a booster and the vaccine. They found in those who are 65 years of age or older, we need 481 persons to be vaccinated with a booster dose to prevent 1 hospitalization. Whereas 50 persons are only needed to vaccinate in the primary series to prevent 1 hospitalization. So the effect of the primary dose is much more than a booster. So the focus has to be on the primary series. Among a younger age group where the disease is milder, you need 8,738 persons to be vaccinated to prevent just 1 hospitalization. Whereas only 396 persons need to be vaccinated in the primary series. So primary series is most important rather than boosters.
Again, Vipul, this is somehow I am a vaccine skeptic as far as the COVID vaccine goes. So I'm asking this question. We've not been able to develop a short vaccine for influenza as yet. That's also mRNA virus, just like the SARS-CoV-2 virus. So what chance do we have of a universal coronavirus vaccine in the near future, which will give us great immunity or great protection, let's say? Flu vaccine to bana nahi humse.
I think we have discussed this point in depth in last couple of questions. I don't see that. What we are aiming at. Are we looking at a vaccine which will stop transmission? No. We should stop looking at that kind of a vaccine which will stop transmission. No, community transmission of this virus is not going to stop. We should accept this for the future, that this virus is going to stay with us. It is going to become endemic in some time and maybe sporadic in long term. So we should stop believing that this is going to go. No, this virus is not going to go. This is going to stay. Yeah. Now, second question comes, if some mutant comes, which is highly virulent like Delta, then what is to be done? Yes, we have to prepare the machinery for that. As Dr. Chugh said, we need to have vaccine system ready, which is plug and play kind of. As we report that virulent strain, we should be ready with the vaccine within maybe couple of months. And highly areas where you have high density of population, like metros and countries which have high influx of foreign tourists or something, there should be.
Go ahead.
They should be basically started with the mass vaccination in that phase. That is going to be the future. And in this, I think there is a next slide to it. Next. In this, subunit vaccine is going to play a significant role in future. And you might have multiple protein subunit attached to nanoparticles, LNPs, lipid particles attached to it, which can be upgraded as and when needed in case of a sporadic outbreak of that specific virulent strain. That is going to be the future of this vaccine or this control of this disease. Rather than assuming that every year we're going to have some new vaccine for this like flu. No, I doubt that is going to be the case in future for this disease.
Okay, fine. So, Gaurav, what are the new COVID-19 vaccines on the horizon, especially for India? I mean, are we going to graduate beyond Covishield and Covaxin?
Hopefully, yes. The government has already given emergency use authorization to vaccines, but there's no word on when they will be launched. So next slide, please. So we've this vaccine has been alluded to earlier in this talk as well, that there's a DNA plasmid-based COVID-19 vaccine, which is already been given approval, emergency use authorization by the government, especially in pediatrics. This was the first vaccine to do pediatric trials, the Zydus Cadila DNA plasmid-based vaccine. These are safer, and next slide, please. But they need a transdermal injector to be used to administer, and it adds cost to it, and it's a 3-dose vaccine. Next slide. It's already been approved. Sir, next slide. Approved in children 12 to 18-year-old. As far back as July 2021, it got approval from the Indian government, and it has shown good efficacy as well against severe disease. And it's an Indian vaccine, and the trial also was conducted in India. Next slide, please. But the most awaited, I think, is the Novavax COVID-19 vaccine, which is being manufactured by Serum Institute. And apparently they have a lot of doses already ready under the name of Covovax. This is again a recombinant nanoparticle vaccine developed by Novavax in association with the Coalition of for Epidemic Preparedness Innovations. And the efficacy reported is above 80% and almost 90% after the second dose. It is stable at our usual refrigerator temperatures. Next slide, please. And again, it got approval to extend the trial to 12 to 17-year-olds. And as I said, Serum is manufacturing it. Next slide. And it's already received emergency use authorization in December of last year. Next. And they are now extending it to children under 12 years in the trial at least. So the vaccine which is supposed to change the scenario world over is the one which is known as Corbevax, and it has been developed by Texas Children's Hospital along with the Baylor College of Medicine in Houston. And basically it's a patent-free vaccine, so they have offered it to the world to use it without any patent. Diagrammatically or diametrically opposite approach to what Pfizer has been doing to the world. And it is supposed to be once it kind of gets into the system by the end of this year, it is going to be very, very cheap, and it should be a lifesaver for the poorer countries. And for us, another very exciting vaccine platform is the nasal vaccine being produced by Bharat. Next. So as Puneet said, it's a, sir, next slide. It's a viral vector vaccine. It's an intranasal vaccine. So it is supposedly, it's expected to give local mucosal immunity, IgA, as well, mucosal IgA, as well as the neutralizing IgG and the T cell responses with the other vaccine, which is hopefully which will prevent transmission of infection as well. Next slide. And it's based on the chimpanzee adenovirus viral vector, where the a stabilized spike protein is introduced into the viral vector using gene coding and then given intranasally. Next. And it has been shown to give protection in mice and hamsters, but still there is no data available in the public domain, and we are just hopeful that this will be effective and will be available soon. Next slide. That's
And anything else, Puneet?
Universal COVID coronavirus vaccines are being worked upon. I think Puneet is the best person to answer that because he's done a review article along with Dr. Vipin Vashisht on universal coronavirus vaccine. I think that would be the ultimate goal to get rid of all these variants that keep.
Yes. Puneet, can you comment, Puneet, on that?
Yes, sir. Regarding universal vaccines, yes, their work is already going on. There are more than 20 groups who are trying to do that. Their different approaches are being followed. One is chimeric vaccine, where instead of encoding for one spike protein, they are adding bits of all the different spike proteins in one vaccine. So that is the first approach. Then even spike protein is a big protein. There are many epitopes. When a vaccine based on even spike proteins injected, body induces polyclonal response. It is not monoclonal response. There are antibodies against different epitopes. Among the thousands of epitopes, they are trying to find those epitopes which are common with other coronaviruses, and they have really found some such. They have even studied the germinal center of the draining lymph node of the person who received mRNA Pfizer vaccine, and they have separated out the antibodies which are common to other coronaviruses. One is 2C08 or something like that. And in animal studies, they have found that the vaccine is effective against wide variety of coronaviruses. Even animal coronaviruses they have studied, and the vaccine is effective. So the work is going on. CEPI has even funded it. They have announced $200 million for this year for the research into this kind of vaccine. So there are different approaches they are trying. Like some conserved epitopes in the helix of the spike protein. Those are also common with other coronaviruses. So that kind of work is also going on. And regarding one comment regarding that Zydus Cadila vaccine, another advantage of Zydus Cadila DNA vaccine is it is stable at room temperature for 3 months. It can be stored at 25 degrees centigrade. So that is another advantage of the upcoming Zydus Cadila.
Okay, so now, of course, not the million but the $1 billion question is that is the end of the pandemic in sight? You get these conflicting statements. WHO says something, Boris Johnson says something else. We hope for something altogether different. So this is open to all the panelists and, of course, the audience also, if they have any comments or questions. When is this pandemic likely to end? That's my first question. And the other one is that if made available to the practicing pediatrician, would you administer the COVID vaccines, at least what is available at present in your clinic? Open to all of you. Sir, Dr. Chugh, you can lead. Mute, sir.
Unmute, sir.
So second part is easier. Yes, we will vaccinate. The first part, refer to my first slide, humility. We don't know. But from what we know or what we are seeing and what is happening in this Omicron, it seems that this will end, the Omicron wave will end in a few weeks, maybe 2 months or so. That is a guess being made by many. And as I said, guesses can go totally wrong. In the meantime, another Omicron pop bhi aa jayega, we don't know. But anyway, that's what it looks like. And after that, it is likely to remain endemic and slowly die down, as Vipul had explained so nicely. So probably that is likely to happen. And it will be something like influenza. We may be needing different vaccines every year or maybe at different intervals. That's my comment.
Seeing the mutations Omicron had, it is getting weaker and weaker. So I think it is going to extinct, to be positive.
पता नहीं sir, बड़ा
We
smart
all
virus
hope so. We all hope
smart
so.
virus है.
Yeah.
It is lulling you into a false sense of security and then it will hit back.
But if I can,
Of course, we are
a
hoping.
virus pops up and in less than 2 months, it has infected millions and millions of people. And by March end, it is expected to infect 50% of the population, despite all our efforts, all our vaccines. I think coronavirus is making new rules and is ruling the roost, so to say. We are only seeing things unfold, and I don't think we have anything under control as of now. And if it ends, if it decides to end, it will end. We are not kind of in control at all.
So just as this cartoon says, the COVID or the coronavirus is here to stay, that might as well clean up its mess. जियो और जीने दे, तू भी जी और हमको भी जीने दे भाई. So really before the vaccine
And
also.
as far as the using vaccine in clinics, sir, I think if it is freed from government control, I don't have to answer to any district government official. Then only I would be willing to use Covaxin. Covishield, I have still many reservations after seeing some near and dear ones going through life-threatening adverse events. I am a bit skeptical of using, but
Actually,
Covaxin
that was the reason why I asked this question, that it's not as non-reactogenic or as safe as the other pediatric vaccines that we use blindly. हम लगाते हैं और बच्चे को भेज देते हैं कि जा भाई. But with the COVID vaccines, a significant proportion has immediate effects, and in the next couple of days, I mean, you'll be pestered by calls from parents. Hello, that's my sense. So I would be
कॉल
slightly.
हैंडल कर लूंगा मैं, बट जो वो इमीडिएट लाइफ थ्रेटनिंग इवेंट वाला जो रिस्क है, I cannot handle that. Covaxin I would be willing to use once I am not answerable to any government official that किसको लगाई, कब लगाई, क्यों लगाई, कहाँ अपलोड किया, वो जब खत्म हो जाएगा then I might use it. But and once Covovax and all these come, maybe if they are safer, and maybe then once we are allowed, that is.
Yeah, thoda sa I would be. Of course, that's my personal opinion. But
sir, मैं एक बात कहना चाहूँगा.
बोलो sir, बोलो.
We had an excellent scientific discussion on the vaccine. All aspects, every millimeter has been dissected very nicely, very well. I think कुछ छूटा नहीं है. But science and commerce are two exactly simultaneous limbs. और आपने जो last question open to all में भी उसको ऐसे छोड़ दिया, उसको ऐसे नहीं छोड़ेंगे. It has got many aspects. एक जो गौरव ने थोड़ा टच किया, लेकिन दूसरा जो फैक्टर है कि कोविशील्ड has to be used in the same session. उसको अभी जो present formulation that is available. So most of the private sector people who were being offered Covishield, they were actually going into losses. Or Covaxin has got a shelf life of, they claim, 28 days. So maybe marginally they are not going into losses. Barely the cost is being covered. So why is the, and you know that the country, आपने भी कई बार कई references में million और billion word use किए हैं, but वो cases और उन चीजों में करे हैं. Million and billion rupees have been spent on COVID by the country, by the taxpayer. But where it has gone, ये बड़ा मुश्किल सा सवाल है, but obvious answers आपको नजर आ जाएंगे. But in any case, the medical profession has suffered the most. और even जो government regulations आ रहे हैं on everything, कि इसका pricing ये होना चाहिए, इसका pricing वो होना चाहिए, मतलब association has to pick up a voice for that. Like अभी जो COVID RT-PCR भी 300 का cap आ रहा है. How is it possible to give a quality report with so much of paperwork, so much of, मतलब एक बंदा तो data entry operator पूरा दिन तो उसमें उसके data और आधार कार्ड और चीजें ही मैच करता रह जाएगा. So how can you test it in 300 rupees? So similarly, the costing of the vaccine, वो vaccine की cost पहले ही cost करके, and it is being projected कि इतने की vaccine है जी, and फिर आ जाएगा डॉक्टर चोर है जो इतने पैसे ले रहा है. और जो Covaxin वाले भाई साहब एल्ला साहब and his wife have been honored with भारत भूषण, वो पद्मा भूषण yesterday.
दोनों को मिल गया सर, उनको तीनों को मिल गया.
तो वो एक को देते तो दूसरे को जूते नहीं पड़ते क्या? तो दोनों को ही देना.
So but मतलब association at some level, I think people, our members, they look towards the association for help